So, welcome everyone to today's webinar. We're going to talk about FAIR in biomedical research today. And before I'm going to introduce the speakers, I'd like to say a few words on GHGA, and I would also like to take the opportunity to announce the next upcoming webinar, which is a beginner's guide to Galaxy, which is going to happen on the 28th of February. And if you are interested in that topic, you can either sign up with the QR code you see here or go to our website, ghga.de/events, and register there. For all of you who haven't heard about GHGA yet, we are the German Human Genome-Phenome Archive. And our mission is to promote data sharing of humanomics data in a GDPR-compliant manner. And for example, taking data from clinical settings to make them usable for research purposes, and hopefully that also will lead to translation of research insights. And we're not doing that alone. There are diverse communities involved, like computational biologists, geneticists, clinicians, physicians, also patients. And if you would like to learn more about that, go to our website. Now I'd like to introduce the speakers of today. So as I said, the seminars today would be on FAIR data, and we are going to have two speakers. One is Sven Nahnsen. He studied biomathematics and biotechnology at the University of Greifswald and did stages in Strasbourg and Cambridge. Continued to do a PhD in bioinformatics in Tübingen, and is now the director of Quantitative Biology Center at the University of Tübingen, and is also professor for biomedical data science in Tübingen. In GHGA, he's the spokesperson for the metadata workstream. And he's going to give you an introduction on FAIR. In the second half, which is going to be presented by Sven Fillinger, you get a more deeper insight into the implementation of FAIR. And Sven is coming from a different direction. He did a master's in plant molecular biology, so a wet lab scientist by training, University of Hohenheim, and then continued to do or be a PhD candidate at the Center for Plant Molecular Biology in Tübingen. But since his heart is beating for bioinformatics, he switched gears and did a master's in biomathematics and bioinformatics and computational biology at the University of Tübingen, and is now the team leader of IT and software development at the Quantitative Biology Center in Tübingen. And with no further ado, I hand over to Sven for the first part.
Thanks very much, Nicole. I have to say I'm always impressed by the outreach activities that are going on in GHGA. I think this is looking for examples. That's really great, and it's a pleasure to together with my colleague, the other Sven, to talk about FAIR in biomedical research today. And we kind of split our endeavor for the next 40 minutes into two parts. So I'd like to give you like a higher level overview on what is FAIR and then what can it be used for. And also I'd like to give you some thoughts on where all this can go. And I'm really happy to also engage and interact with you. So please speak up either during the talk or after the talk in a discussion. And then Sven will illustrate how it can be done on a lower level, how the implementation can look like. So let's get started. I mean, talking about FAIR, I think we should start about introducing the four letters. So obviously FAIR stands for F for findable, A for accessible, I for interoperable, and R for reusable. The acronym has been coined not too long ago. I think it was a paper in 2016 where Wilkinson and colleagues basically published the idea in a perspective sort of article, and basically international communities took on the acronym with the idea of research data needs to be verified. So we need to make sure that we have tools and infrastructure available. So that's one side, but also we need to have the mindset at researchers to really make research data available in a way that colleagues all around the globe can find it, that they can access it in a way that it's allowed. Now, dealing with human genomes, it's of course not uncontrolled access, but controlled access. So fair does not mean that everyone should have access to any data point, but there needs to be like a clear defined access mechanism. It needs to be interoperable, meaning that whatever happens in Tübingen, what data is produced there, published there, that of course needs to be interoperable in a way that other people can read that data, that machines can understand it, that humans can understand it. If we do like the first 3 letters well, then of course our data is reusable. And then we can really start doing big data research in a way. Now, why biomedicine? I think the corona pandemic that we all suffered from quite a bit in the last 2 years, 3 years, is really a success story in biomedical research, because obviously there was this virus. We could with instruments, sequencing instruments, we can really identify every single base of the genome of that. And this led then to the design of a vaccine that helped us to control the vaccine. Now, this only worked because we had access to the sequencing data of the SARS-CoV-2 virus, and we could like integrate many different genomes all around the globe, and only then we could really have consensus understanding of the genomes, and could of course also identify, I mean, we in the sense of the scientific community, right, globally, we could identify the conserved regions that are also biologically relevant, and that led the companies to produce a vaccine that specifically targets or stimulates the immune system to target specific regions, and as I said, allowed us to control the virus. Now, this is one example. There are many other examples, and we're still awaiting like the success stories in cancer, for example. And we indeed hope that GHGA and the verification of human genomes can actually make a difference there. And at some point, we hope that we can also like report success stories on that side as well. Now, obviously, there's a lot of data out there, and not all this data is fully fair. Yet, if we make attempts to even verify it further, I think there's a huge potential for science. For example, I guess many of you have been looking to the very nice portal, Our World in Data, where you have like a lot of data, like life expectancy is just one data set. So we have a lot of data on the population level concerning some biomedical research, and you can read out interesting things. On the other hand, you have like, we can identify with this population scale data, what are the causes that we die of, etc. Now, this data is fully accessible. It's open data. It's easy to access. It's easy to use, but admittedly, it's low resolution. So it's really hard to really extract scientific facts from these population scale data. But it is there. It is accessible, and you can find it. I mean, not in the sense as Sven probably will introduce it later on, but once you know the location, you can find the data. It's interoperable if you go with the API. It's like test-based data, so you can also browse it. It's a defined schema, and of course you can reuse it. But as I said, low resolution. Now in molecular research, I mean the omics data set, so looking into DNA information in DNA molecules, RNA molecules, proteins, metabolites, etc. This has basically been triggered, or is at the forefront of bioinformatics and molecular research since probably 2 decades or so, where we get very detailed information. Just to illustrate here, we can correlate, for example, protein with transcript expression and then deduct information from that. Or on the right-hand side, you find one figure from a very interesting series of paper where colleagues looked into pan-cancer, so many different cancer entities, and identified single mutations that seem to be drivers for specific cancers. So this is very high resolution, right? We can look into our 21,000 genes. We can look at 30,000, 40,000 transcripts, and we can look to a good share of the proteins that are expressed in a single cell if needed. So this is very high resolution. It's only partially open though, so it's not easily findable. It's not easily accessible, and it's certainly not interoperable, and that's why it's also only partially reusable. Well, partially open because it's occasionally human data, and not always we have the ethical and legal allowance to make it fully open, etc. And it misses still metadata definition, so it cannot. It's not fully interoperable. So there's still lots to do for making like this high resolution molecular data fully accessible and fully fair. The metadata is often not standardized, and like this does not necessarily relate to fair, but it also requires a significant expertise to process the data. Typically, I mean, careers of bioinformaticians went into processing software applications to understand, say, DNA sequencing data or RNA sequencing data, which means that we cannot easily and fully integrate these different omics layers, plus even integrated with population scale data. Now, what's next? I talked about population scale data that was fully open and fully accessible, yet low resolution. Then we had looked at high resolution omics data, and there's more data that is available out there that is not fully used in research yet because it's not fair. There's of course a lot of imaging data on the left. You see some results from our own research where we can basically find tumor regions in MRI images from radiology departments, and with machine learning you can then find which regions are tumor regions, which are not tumor regions. There's significant, there have been quantum hypes in the way how microscopy is being performed in the lab. So we can have now multi-layer microscopy where we can label dozens of proteins in Codex or Maxima instruments, and we have a lot of data. So this is really high data. So this again is only partially open because it misses like clear routes to really make this data fair. And occasionally it's also human data and cannot be fully anonymized. It has more and more increasing resolution. So we can now do that at the single cell level easily in microscopy without major investments. So I think imaging will really deliver a significant share of really high quality research data soon. At the moment, it still, again, requires a significant computational expertise. I'm putting quite some emphasis on that because if you want to integrate imaging, omics plus population scale data, and if one needs to be expert in a single domain, that of course also limits the integration ability somehow. And again, metadata standards are also missing here. So there is a lot of data, and I think it's super promising and super exciting what can be done theoretically if we integrated all that data literally for humanity, right? There are so many diseases that we have a chance to understand better, and they also then think about cure for them. So there is a lot of benefit, but as I said, we need to work on making these data fair to really find all this benefit a lot. So I think I want to bring the message that there are deep cracks, so to say, in the biomedical data landscape. We've been looking to imaging data, we've been looking to population data, we've been looking to patient data, clinical studies, and then there's the whole range of molecular data that is partially from fundamental research, that is partially from patients. There is a bit of repositories that are building like globally. There's of course the reference data sets that are being used for bioinformatics tools to find mutations, to find differentially expressed proteins, differentially expressed transcripts, and so. And so there's reference data, there are repositories. However, to integrate all that and to really benefit from all that, it's not easy because you need to jump over the deep cracks, and this is not really happening. Just to illustrate a bit more, imaging with respect to repositories, there are the global initiatives. There's a lot now being also on the commercial cloud applications, which again requires computational expertise that is not necessarily given. So you need, there's a lot to learn, with the if you attempt to integrate all of that. Now, our research at the Quantitative Biology Center here in Tübingen is partially to bring these data sets in shape. So we want to understand them. We want to be able to process the individual data types in a standardized fashion, and we want to also have we want to understand, and with I want also say machines need to be able to understand metadata models, and this needs to be standardized in order to fulfill the integration endeavor and thereby generating or working towards the potential that is in these data sets. Now, to do that, of course, we need to start to modeling the data. And then this is now a very high level view on the metadata model that we developed for human genomes. And as you can see in the pink slot, there's the data set, and there's of course a lot of surroundings. And these are the descriptors that we need to have for the data. And this is really the prerequisite to verify the data, because only if we as a community, and I really mean a global community, agrees on what needs to be there to describe the data in a way that it can be findable, that it can be accessed, that it is interoperable. So metadata is all about interoperability, and that finally we can reuse this, that we can integrate it with other data sets where we have similar understanding of the metadata, and that we can have really large-scale data sets, because I think most of the people that do data-driven research are aware that the more features we have, and obviously the genome has a lot of features, the more data sets, the more samples you also need to integrate in your study to really reduce causal relationships at the end. At the end, we obviously want to do that we can also efficiently answer questions in biomedical research. And this is just a question that really arose in our own research project. So we were approached by our oncology colleagues with a statement like, well, my mouse model suggests there should be amplifications on the sex chromosomes for a certain set of genes. And that in my mouse model seems to correlate significantly with a certain cancer phenotype. And now this is mouse model, right? Can you help us somehow if this is really true in human? And luckily, we had access to a large amount of data from these public repositories. So we could say, yeah, hang on, give us some time, because we had reproducible pipelines, and we could really identify the publicly available genomes that are relevant, so that are basically had the relevant cancer entity. In this case, it was hepatocellular carcinoma. We could identify these data sets and run our standardized workflows and find then the correlation. So we could answer that relatively quickly. And so like many decades back, one would probably have to run a clinical study to answer that question. Now with publicly available data, that is fair to a certain extent. You of course have a chance to answer these questions. Now, questions and data, you probably, I guess we all have heard about ChatGPT lately. So the large language models that give us answers, whether they're true or not, to questions, but you get sound answers. And this is, of course, something, and here I brought you an illustration on how you can basically get a burger recipe from ChatGPT. So this works well. And there are, of course, also cases where it doesn't work well, but I will not judge on the quality yet at the moment, but you can, of course, get answers to questions. So this is, of course, something that we'd love to also have for such biological questions. And let me just go back. The biological question here was, is there an amplification on certain genes on the Y and X chromosomes? And if you asked at the moment ChatGPT such a question, it doesn't work, because of course it's not encoded in the text-based data, in the internet data that was there to train the algorithms. However, if we do a good job in the verification of our data sets, I think there is huge potential in having such tools then run and having hopefully good answers to it. Now, before we go to the lower level, briefly back to biomedicine, how do we practice biomedicine at the moment, or there's of course the one part where we want to organize knowledge and then make it accessible. So we call that the knowledge-based approach, where data science and like fair data can really do a good job. And then a bit the future is, of course, that we want to have predictive assessment of data and really use existing data to predict for an unknown case what comes out. Now, for both routes, the verification process is really essential. Let me briefly talk you through the two things. So firstly, the knowledge-driven biomedical data science approach. This is literally all about FAIR. So information is typically recorded in central databases. Standardized description make information findable. And the stringent use of metadata is, of course, essential to have it findable. This typically, I mean, biomedicine is frequently still performed in silos, so either in specific institutions or if done well, sites integrate the data already, but we don't have a global integration yet. Of course, you can also make locally your data findable. Then we need to develop interfaces to also build a bridge between the application domain and the data scientists. So interfaces are required to make the data accessible. For biomedical data, frequently access restrictions need to control access. If, of course, one needs to monitor the ethical and the legal constraints, then the data should not be bound to a single environment. Using community standards, we want data from site A to be usable for site B. In other words, this data needs to be interoperable. Finally, as I mentioned, if we implement F, A, and I with a good knowledge base, and we can actually use that knowledge base to efficiently make use of existing knowledge. We don't need to basically do everything again from scratch if that has been performed before, because we can look into databases like typically SQL database and look up the information and then perform accordingly. So this is what we then call knowledge-based. So this can work like in typical molecular tumor boards, for example, that you have cancer genome data, you have the patient metadata that encodes like the tumor entity and other metadata. Then the clinical colleagues come up with treatment decisions and an outcome. And then, of course, if that is collected in a large resource for the next patient, you can look up what was there a similar profile, and if yes, how was the treatment decision, and can we now decide accordingly. And of course, the larger that knowledge base is, probably the more experience can go into the upcoming decision. So this is still like manual decision support. It's a knowledge base where people can look up, and this can lead to still already very nice visualization. So this is illustration again from the pan-cancer genome series of papers where the colleagues could find specific mutated genes in a large variety of different cancer entities and could then find out which genes are most prominently mutated. And this is a knowledge base that is there, and it's very, very, very important. That can, and a lot of research can already be done by that knowledge base. However, it's not predictive yet. So what we think fair data can also help with is that if we build a knowledge base correctly, so again, we collect, for example, cancer genomes, we collect the patient metadata, we collect the treatment decision, we collect the outcome, then, and if that is collected in a fair manner, then, and if it's huge, then because FAIR also allows us to integrate data from many different sites, then in the future, hopefully for an unknown cancer genome data with, we can then use machine predictions, obviously, and we can have a truly decision support system. We can come up with prediction for the treatment, potentially prediction for outcome. This, of course, can be collected again in the knowledge base, and then we can also for the future align the accuracy of the predictions, and hopefully this gets better. So I think this is a great potential if we make sure that our data is really fair, and we and certainly many others are working on making that reality. So maybe I'd like to conclude like the first part, which is the bird's eye perspective. What we want to do with the FAIR data is that we want to use it to predict some outcome for the unknown input. It's very likely for a cancer therapy decision support system. And we need to obviously work hard on interfaces, because only with good interfaces we can firstly build the bridge between the application domain, which is in this case oncology, and the data scientists. And we can also facilitate the access for machine learning methods. In any case, whatever is happening there, and that's really a research area on its own, the predictions need to be robust, safe, and reproducible. That might be another topic for a talk. The data ecosystem that we build, if we do it rightly, is of course ever growing. New prediction outcome pairs are being fed back to the system, and as I said, we can then monitor the accuracy. And then if that's of course always working in the interaction with the application domain and with the data scientists to build clinical decision support systems. And I think this vision is basically building on fair data, and this can only work if the data is fair. And I think with that, I'd like to conclude like this motivation introduction part, the bird's eye perspective, and I'd like to hand over to Sven Fillinger now. Or Nicole, do we have questions directly, or shall we?
Yes, maybe before we start with the second part, I would like to open it up to the audience to ask some questions. If there are any, please feel free to either post it in the chat. I haven't seen any yet, but there is still time. So maybe one quick question from my side. We've heard a lot about the benefits of FAIR and data sharing. So how do you deal with all the people who still doubt it, for example, when it comes to data ownership? Does that repel people from trying to be fair?
Absolutely. I mean, there are a lot of benefits, and I think we're on a good track also to kind of convincing our colleagues that are still reluctant to do it. I think we should also, like we as scientists, we also need to produce these showcases where really we can generate additional, really scientific, high impact science with fair data. I mean, they're like the Peacock papers, they were already super showcases. I think we need more of those. With respect to data ownership, I think in turn, we could probably have some better ideas on how we can also give some credit back, to give more incentives, for example, that we have like applications where we have a data provenance mechanisms where it's really tracked from whom comes data, and to also like have more acknowledgements and potentially also authorships for people that verify that data and make it really available. I mentioned that in various times before. I think the physicists occasionally they do better than the biomedical scientists, because I mean, physics has seen big data earlier, and they have been dealing with like these global big data type of research settings. And I think they were not so reluctant with it. But if you also look to the physics papers, they have hundreds of authors on these papers that really dealt with these big data. And I think we can do better there as biomedical scientists. So that would be my plan, to improve on that side, despite the fact that we, of course, need to convince our colleagues that this is the way to go, and that we really need to integrate our data sets to do big data science.
There's still a long way to go. We have another question in the chat. So this is from Mohammed Al-Medavar. Can standardization of metadata be done on existing databases, or is it a pipeline developed for planned data collection activities?
Yeah, I mean, typically it's easier to do that on a prospective setting, because then you have a white paper, you basically bring the community behind your metadata model. However, you can also like do metadata modeling for retrospective data. You then probably need to look at how is the data annotated at the moment. Is it possible to to fill the missing properties? Can we convert it even automatically? It will not work for all data sets, but there is a chance that you get a good share of the historic data still integrated, but of course not every data point can be verified from historic data, but prospectively it's easier.
And we have one last question here, and this is something that really keeps us up as well. How to make fair data comply with the requirements of data protection, for example, GDPR?
Yeah, I think that is work in progress, and that really, I mean, I guess I'm speaking for all GHGA colleagues as well, that really turned out as one of the major research components that we really moved forward there. But as I mentioned initially, so the A in FAIR does not mean that if you verify your genomes that everyone needs to have access to it. I think that's occasionally also a misinterpretation of the acronym. There can still be some sort of data access committee, but there needs to be a mechanism how you can theoretically get access to the data. That does not mean that every data set lies in the internet. So I'm very, very sure that FAIR will be possible, or it even is occasionally for human data. So that is being solved.